Just a little bit of what I'm going to talk about. I'm going to discuss the history of thymectomy for myasthenia. And I think this is maybe a little bit of a scary topic. This is the way knowledge develops in medicine, not just in myasthenia. I'll talk about the MGTX trial. Don Sanders yesterday considered that one of the most important trials in the history of myasthenia gravis. And I'll belabor a lot of detail there because I think it will give you an idea of the complexity of how a clinical trial is done. I'll give you the results in a very abbreviated fashion. We too are embargoed by the New England Journal of Medicine, so in a couple months you'll get those press releases. And then I'll just briefly talk about some special aspects of the MGTX trial that we took advantage of in my laboratories, starting to take advantage of. So this handsome fellow here, Alfred Blaylock, was the first individual to perform a thymectomy specifically for myasthenia gravis. He did this in a very few patients. This is his first patient. You can see the before and after shots, significantly better. And she had been coming in and out of crisis at the time, and 2 years later, after a thymectomy, was doing wonderfully. He went on and did a few more patients in 1941. Just please note the differences here now. He had moved from Vanderbilt, where it was a very healthy environment, to Baltimore, Maryland at Johns Hopkins, took up smoking, and the rest is history, and stopped doing research in myasthenia. But he made a very important statement. We wish to emphasize again the absence of conclusive proof that the improvement noted in our patients is due to the removal of the tumor from the thymic region. And for decades after, people, physician experts, have been confused about this. This may be a little difficult to look at for those of you not into graphs, but let me just summarize this. In this study in 1990 of the MGFA MSAB board, basically, up to 2/3 of the individuals surveyed had reservations about sending patients to have a thymectomy. And there was a middle group who sent a few patients, 1/3 or 2/3, and then up to 10% who just did not send patients for thymectomy. So a lot of questions about this. And then Gronseth and Barron did an outstanding evidence-based review of all the quality studies of thymectomy. These were all retrospective. They identified major shortcomings in nearly every one of them. So not really a good situation when I'm a physician talking to a patient as to what should be done. And their recommendation here was a control trial is needed. And that's what's done. So John Newsom Davis, the late John Newsom Davis, he died in a tragic car wreck and after the trial was initiated. And Gil Wolfe came together and organized the MGTX trial, along with Fred Jaretsky, who also started with us in his mid-80s, made it into his 90s, never saw the results of the trial. And I'd say, most importantly, the team at University of Alabama, Birmingham, which without them, this trial never would have happened. Absolutely. Because we failed a bunch of times until they came on. As you can see, we had our first organizational meeting in October of 2000. We finally got funded after 4 tries to the National Institutes of Health. The equivalent European funding body said no to us, and we didn't try again. We had an investigator meeting to teach everybody what to do. And you're going to see what the complexity of this was. We got everybody in 2 sites, Oxford, England, San Francisco, California, obviously awful places for people to go to, but they went. and learn about how this trial was going to run. The National Institutes of Health at that time appreciated this is an opportunity that we can collect specimens from patients to understand the biology of this disease better, and they funded us to do that as well through internal processes. We got our first patient in fall of 2006 and finally completed in 2015. So this was a 3-year study. So what did we try to learn here? So these are our basic questions. Could thymectomy plus prednisone versus prednisone alone guarantee greater improvement in weakness? Could we lower the dose of prednisone, and could we reduce adverse effects in either one of these conditions? We didn't have a bias. The investigators who started this basically said, I don't know and I want to find out. And we had people who said, no, I'm not going to do this because you're nuts. Thymectomy works. We had people who said, you're crazy. Prednisone and all the great immunosuppressants work. I don't send people to thymectomy. That's the perfect situation for a clinical trial. That's the way it's supposed to be. People don't know. Patients don't know. They need to have better understanding. Physicians are obligated to do these. What kind of thymectomy procedure we did? We knew we had to standardize this. Fred Jaretsky had done extensive work to indicate how each of the surgical techniques was very different in how much thymus tissue was. If you're going to subject yourself to a surgery, you better get this stuff out of there. We chose the circled one there, a transsternal thymectomy. Many of you have those scars. This was initiated just at the beginning of videoscopic robotic procedures, so we chose this procedure where we could get the most surgeons who are familiar with this. And oh, by the way, black here is better. We didn't go for the most extensive procedure because that was more dangerous. We had, as you'll see, surgeons from across the world. That's going to vary in their expertise. And if they hit a phrenic nerve, your diaphragm is gone. We didn't want to have those things. If it were Fred Jaretsky doing every surgery, then we would have done that, but that's not possible, and it's not real life. So who do we study? This is not MuSK patients, this is not seronegative patients, this is a certain group of individuals, but it makes up the vast majority of individuals. They had to have acetylcholine receptor antibodies. Their disease duration was relatively short, 5 years. There was a bias to feel that thymectomy may not work later in the disease. Started at age 18, went to less than 65. That's the population. And you had to have more than ocular, but we're not going to operate in crisis, but you had mild to moderate to severe disease. That's what that class is. So how did we do this? Well, first of all, this was randomized. There's a flip of a coin, and this is where the subjects come in. They were willing to have us flip a coin between a surgical procedure and a non-surgical arm, each getting prednisone. This is one of the few surgical randomized studies in the history of medicine, and we owe it to the subjects for doing this for us. Blinding. Well, obviously the patient knows whether they had a surgery. We accepted that. And nobody thought it'd be a good idea to do a sham surgery. So we had a group, we had a treating investigator at the site, and then we had a blinded investigator. Or, as the National Eye Institute prefers, masked, not blinded. And that blinded evaluator wore a beautiful NIH-produced turtleneck to hide a scar or not. And at 3 months, patients would have recovered, and they get tested by this blinded evaluator, and then their regular care is provided by another investigator. The importance of that is we don't want some bias to be entered on either part. And so, as it turned out, when we evaluated our investigators as to where they thought this was, they were at random. The blinding worked. They didn't understand who was getting surgery, who was not. And then we did follow-ups up to 36 months. That's where our centers came from, all across the world. And I'll have another slide as to why that was so important. We knew, ultimately, there weren't going to be a whole lot of patients that wanted to be in this, that could go through 3 years. And then there was going to be a large group of patients, you can't see this, but I'll just tell you, that we screened 7,000 patients. Now, not all 7,000 were into the entrance criteria. There's some ocular patients there that clearly didn't, could not go into the trial. There are people who already had a thymectomy. There are seronegative patients. There are MUS patients. So that 7,000 number is not that impressive. But I'll take you down a little bit further. There were about 200 patients who could have qualified, but they refused. Either they wanted a thymectomy or they didn't want thymectomy. Or they wanted prednisone, or did not want prednisone under any circumstances. That was the biggest kind of exclusion criteria. There were a number who did not want to be guinea pigs in a clinical trial, despite fulfilling the inclusion criteria. They got randomized, 126 of them. I'll just tell you that number is statistically significant. So we had to recruit to a certain level to assure ourselves of a meaningful result, and we got there because the subjects kept with it for 3 years. They did not give up. The investigators did not give up. It's amazing. Now, here's the confusion. Each of these 3 columns represents potential answers, and there's more, but I'll just kind of summarize. If you look at the central one, prednisone is similar in each group, same strength, same adverse effects. Well, there's no reason to do a thymectomy there. A mixture of results. Well, if patients are stronger, but they're on high doses of prednisone with high adverse effects, well, that's not really all that great. And you can think about that in either arm. Or if the complications of surgery are so severe that the adverse effects are so bad, even if there's a benefit, you don't want to go into that, right? Now, I can tell you nobody died because of the surgery. We had extremely low complication rates. It was fantastic. And oh, by the way, Fred Jaretsky made every surgeon take a test. If you know surgeons, they're never going to agree to what a neurologist said. That's why we had a fellow cardiothoracic surgeon submit the test. He looked at every surgical report. We had each thymus photographed and weighed. He looked at it to make sure these men and women surgeons were not screwing up, and they were doing what we were asking, and there was a huge compliance rate. So the next slide shows the result. So unequivocally, we found that thymectomy works, and it works pretty quickly. This is not a cure. This is not a cure, but it lowers the prednisone dose, and it makes people stronger. So there we go. Something that never could have been said before. Now, as I said this is going to be very brief, and there's going to be some technical slides here that you don't need to worry about. But I think it's important also to appreciate what the MGTX trial is going to bring into the future. So in addition to all the things that were being done, and the close follow-up and the examinations being done, each collection site from across the world shipped these biofluids, so your blood tests, the thymus, in various different fashions. By the way, some of these things came in from Australia, Argentina, Chile, It was phenomenal. The guy in Argentina had to pay off the customs officer in order to send them off. That was not budgeted and would not have been allowed by the National Institutes of Health, but he got it done. And we had rigorous methodologies. Every kit came with specific instructions on how to ship. Oh, by the way, you can thank World Courier and FedEx because they also repacked some of the dry ice or ice to keep these things going as they came through customs. The thymus, each site had to follow a strict protocol in terms of obtaining tissue in 2 different ways. And we have the largest biorepository linked to rigorously defined clinical endpoints in the history. We also have 3 years of clinical data. So this is a natural history study. There's never been a natural history study like this in myasthenia gravis. So now we can tease out what this disease is about. And I don't want to go into this. I'll try to hit the time points. My laboratory with Dr. Kushner, my close colleague, did what's called a microarray and mRNA analysis of these tissues. This is still preliminary. And this is a summary slide of what we found. So that mRNA, microRNAs, that's what regulates physiology, and in particular the immune system. And so what we found is a key area is apoptosis, or survival of cells. So the apoptosis is something that kills cells. So sometimes that is good, sometimes that's bad. When you have a cancer, these cells are growing and can't be controlled. And there's these anti-cell death proteins that are expressed at very high levels. Well, so that's kind of similar to autoimmune disease. You've got a certain small population of cells that persist. They keep on producing these antibodies. That's not the way the immune system is supposed to work. It's supposed to protect us, not supposed to hurt us. They evade elimination. And there's certain checkpoints that don't work, just like in cancer. So we had a postulate that perhaps this was the case in myasthenia gravis. And I think I'm missing a slide there, but those array profiles that I showed you, they pointed to these anti-apoptotic mechanisms, one of which we had been studying already, looking at a protein called survivin. And just in the latest Journal of Immunity, this is clearly a very important protein that directs how antibody-producing cells exist. And pictures are worth 1,000 words, and so if you can turn off the sound so nobody gets blasted. This is a rat, in case nobody's seen things like this. And so this is before our treatment, myasthenic crisis level rat. After, with treatment, doing great. And these other panels, the graph just shows one animal being very strong, one group of animals, the other one getting weaker and weaker. And then this last set of graphs show how we can eliminate the blue dots on one side of that line in the lower panel, and that's where our treatment approach versus the one that did not receive the treatment has these cells that are have this cell death prevention protein in it. So our next step in taking forward all these specimens from the MGTX trial is to figure out how we can eliminate those cells that are causing the disease. We know that the thymectomy now works. Unfortunately, it's not a cure. We got to figure out what's going on outside the thymus, but this has given us already some insights into how the disease works. I think that's my last slide. Well, my summary. One of the few single-blinded studies in history. We've demonstrated an effect, and with many colleagues and hopefully more, will continue to work on this rich sample bank to further discovery. So just some acknowledgments of people at George Washington. Dr. Marks is the world's expert in the thymus. He's been the pathologist in charge of the MGTX study and has characterized these studies. Kevin O'Connor is an immunologist who's doing other studies on the DNA of individuals from the MGTX trials, already identified very interesting characteristics of how these antibody-producing cells expand. I mentioned the key contributions of Dr. Cutter and Dr. Aban, his close colleague. And allow me to add, the biomarker studies, these studies of the specimens, started its funding from the MGFA. Without the MGFA, Dr. Marks would not have been able to do the initial characterization of the thymus glands. The New England Journal of Medicine actually said you have to have this kind of stuff. If the MGFA had not ponied up the money so many years ago, that wouldn't have been done, and paper probably wouldn't be accepted. So the MGFA along the way has made key contributions to this and supporting having a presence for the continued information about the trial and solicitation of subjects for the trial. So my hat's off to the MGFA there.